I wish you all warmly welcome to the 2020 Crafoord Academy Lecture in Biosciences, when we will listen to Professor Penny Chisholm from the Massachusetts Institute of Technology telling us about what a tiny microbe from the sea has taught me about life. Due to the corona crisis, we are this year not able to have the lecture in the usual way in Lund. And that's also why I'm sitting here in my office in Stockholm. But I'm sure this web lecture will do perfectly well. My name is Ove Eriksson, and I'm chairman of the Crafoord Prize Committee in the biology class of the Royal Swedish Academy of Sciences. And before I hand over to Karin Rengefors in Lund to introduce Penny Chisholm, just a few words about the Crafoord Foundation, the Crafoord Prize, and the Crafoord Academy Lecture. The Crafoord Foundation was established in 1980 by the great Swedish industrialist Holger Crafoord. And the main aim of the foundation is to support scientific research and education, but it also supports many other activities, social, cultural, and artistic. The Crafoord Prize is based on a donation to the Royal Swedish Academy of Sciences from Holger Crafoord and his wife Anna-Greta, and from the foundation, the Crafoord Foundation. And the prize has been awarded since 1982, following a 4-year cycle. The first year, mathematics and astronomy, second year, geosciences, the third year, biosciences with special emphasis on ecology, and the fourth year in the polyarthritis. And then it starts all over again. In addition to polyarthritis, an illness Holger Crafoord suffered from, the choice of subjects aims at complementing the Nobel Prizes. That is, in subjects in natural sciences, which, with the possible exception of astronomy, for example, this year, are not covered by the Nobel Prizes. A few years ago, the Crafoord Foundation and the Royal Swedish Academy of Sciences decided to arrange a yearly Crafoord Academy Lecture in Lund, primarily directed to high school students. And the plan is that the subject for the lecture should be associated to the previous year's Crafoord Prize. And as Penny Chisholm was awarded the Crafoord Prize in Biosciences last year, 2019, we could think of no better choice than inviting her to have this year's Crafoord Academy Lecture. And we are very happy to have her here again. So again, warmly welcome to you all. And now I leave the word over to Karin Rengefors in Lund, who will present the program and introduce Penny Chisholm. Please, Karin.
Dear high school students, welcome to this event. I know you're out there in Lund, in Malmö, and in Uppsala and around Sweden. And this event has been designed for you and to inspire you. So I'm very happy to see you out there, even though you're not in front of me. So my name is Karin Rengefors, and I'm a professor at the biology department here at Lund University. And so I have the honor of being the chairperson of this event. And so what's going to happen is that I will give a short introduction about Penny Chisholm, the prize winner, and her field of expertise. After that, Penny will give a lecture. Following the lecture, we will have a short break, and then I will introduce the panel to you, and there will be a panel discussion. And as you've heard, you will be able to ask questions, and I will, in a little bit, let you know how you can do that, so you can send in questions to us. So, but first, an introduction about Penny. Penny Chisholm, and Penny is the first name she goes by, and that's why the Penny in parentheses. She won, as you heard, the Crafoord Prize last year in Biosciences. And that was something I was especially thrilled about, and I will let you know why. And here I will let you in on a secret that I haven't told anybody until now. But Penny is actually my idol or role model. I heard about her about 20 years ago when I was a young researcher. And I heard that she was a brilliant scientist, that she was a really nice person, and I should really try to get to know her because she would be a great mentor. And so I did my homework, I read her papers, and I have followed them since then, but I actually didn't have the opportunity to meet Penny until 2019 when she came to Lund for the prize. So as you understand, I was really thrilled when I heard she had received the prize. And I was especially excited because Penny works in the field of phytoplankton ecology, and that's a field that I work within. So it was lovely to see some attention on this field. So phytoplankton. So most people, when they hear about phytoplankton, they hear about what they cause. Phytoplankton cause algal blooms. And as Swedes, you probably know that algal blooms are really common in the Baltic Sea. And I think that many of you may have seen these images, the satellite image of the really vast phytoplankton blooms across the Baltic Sea that you can really see very clearly every summer. Some of you may have visited the coast at the time, and you've seen this brownish, yucky mess in the water, and you really don't want to go swimming. And these phytoplankton blooms occur in other places as well, around the world, around the coast. And sometimes the blooms are brownish, yellowish, reddish. If you visit lakes, you can also see algal blooms, and it's a greenish mess. You can even see fish kills or other animals that have died. However, phytoplankton are not only bad. In fact, phytoplankton are beautiful. And phytoplankton are necessary for the ecosystem. So let's just take a moment and look at the beautiful. Look at all these different shapes. These are all planktonic organisms, single cells or cells that are in a colony together. They are all photosynthetic, but some are brown, some are green, some are yellow, some look like stars, others look like jewelry. They are very diverse and very beautiful. But of course, most important, they are necessary for the ecosystem. And I like to use this analogy. So phytoplankton form the base of the food web, just like the grass on the savanna. So on the savanna, you would not have zebras, wildebeest, elephants, lions, etc., if it weren't for the grass, the primary producers in the ecosystem. And it's exactly the same thing with phytoplankton. They're there in the sea. Without those, you won't have shrimps, fish, dolphins, whales, everything else. And so that's why the phytoplankton are so important, and this is why phytoplankton ecology is important. So then let's move over to Penny Chisholm. So she was awarded the Crafoord Prize for her pioneering work. And what was that work? Well, the main things about Penny's discovery was that she discovered the most abundant and the smallest phytoplankton found in the oceans. And this was an organism that was later named Prochlorococcus. And if I try to summarize Penny's research, because it's not only about the discovery, it's what you do once you've made the discovery. Well, one of the main things she's been doing is that she has worked with the interaction of Prochlorococcus with other microbes, so that means other bacteria, other phytoplankton, viruses, and also how it interacts with its environment. How is it influenced by temperature, by nutrients? And this is all within the realm of ecology. Moreover, Penny has also worked with Prochlorococcus genome. So all the DNA in that cell, what is it doing? What are all the genes doing? How does it function? Now, you might wonder, why is this discovery of Prochlorococcus so important? Well, as I mentioned first, it is abundant and actually makes up 50% of all the phytoplankton in the sea, despite the fact that they're actually the smallest. And so what does this really mean? So just to give you an idea, I'm showing you a picture of two phytoplankton. One is Nodularia. This is the big thing you're seeing that looks like green filaments. This is the organism that causes algal blooms in the Baltic Sea. And each one of those tiny things marked by the yellow arrow, that's a cell. And one cell is about 10 micrometers, so very small. Now, Prochlorococcus is even smaller. So if you look at the small square next to the nodularia, there are small dots there where the blue arrow is. Those are Prochlorococcus. And they are much smaller. They are less than 1 micrometer. So this is really smaller than anything else. And this is, of course, why nobody had seen it until Penny did. So even though it's small, it's abundant, and it plays a major role in the oxygen production on Earth. It plays a major role in the uptake of carbon dioxide from the atmosphere. Also, in Penny's work, she has been able to increase the understanding about genomes, the DNA in the cells, and also about the genetic diversity in phytoplankton and microbes in general. So the next question left is, why Penny of all the researchers out there? Because there are quite a few. Well, I've thought a lot about this, and I have some ideas. And I think one thing is that she was able to see what others didn't see. Also, she had the wisdom to realize how important her discovery was. Moreover, she had the intuition to study it further, and actually the boldness to focus on Prochlorococcus and not on anything else. And finally, Penny understood that Prochlorococcus have stories to tell us, and very soon she will tell you those stories. So finally, with this slide, this is from Lund, from this room last year. I would now like to give the word to Penny. So over to Boston.
Hello, everyone. It's really very exciting to be here. I wish it was in person, but this tiny microbe, this virus that has taken the whole world to its knees, has gotten in the way of travel. So I'm really happy to have an opportunity to talk to you today. I want to thank Karin for that very humbling introduction. And I will be talking about the beneficial microbes on the planet. 99.999% of the microbes are serving our life on the planet. And I think it's important that we focus on them now and then. So today, as my title says, I'm going to talk about this tiny microbe, Prochlorococcus, but more broadly about the phytoplankton and how studying them has taught me so much, not only about life on the planet, but about life, about how to live your life, about the choices we make. So I'll talk a little bit about how I got into science and then the discovery of Prochlorococcus and where that has led me in my research. So first, I grew up in Upper Michigan on Lake Superior, which is one of the Great Lakes in the northern U.S. And I just sort of took lakes and water for granted. I was there through my entire childhood until I went away to college. And then when I got to college, I majored in biology and chemistry, but I was able to do an independent study project. And that was to study a tiny lake. It was called Round Lake. And what I did was study the manganese budget in that lake for no particular reason, just because it was interesting to learn how to measure manganese concentrations. And we measured every week the concentrations in the streams going into the lake and coming out of the lake. And we had to map the physical size of the lake by going around in a boat and making soundings, and we made a map, and we were able to measure the seasonal changes as the manganese exchanged with the sediments and calculate sort of the life of manganese in a lake. And this is when I learned about science as a way of knowing something. We actually published a paper from this, and it made the lake come alive sort of as a being, not just a big pot of water, but a being that had a life, a seasonal change. And that was very exciting for me. Also, it made everything in the textbooks come alive. Suddenly I could measure what the textbooks were telling me was happening. But then I looked under the microscope and I saw the phytoplankton. And Karin has already shown you some pictures of these beautiful, tiny, we call them plants, but they're not really related to plants, but they're single-celled photosynthetic, well, they're distantly related to plants, photosynthetic microorganisms that dominate every body of water and even grow in ice. Wherever there's water, you will find phytoplankton. And as Karin said, they're range in those days, they ranged in size from maybe 10 microns to 100. Human hair is 100 microns in diameter. So just to give you a size reference. So I studied phytoplankton as a graduate student from lakes, and then moved on to the oceans as a postdoc and on to MIT, always inspired by these tiny microbes that are generally unappreciated in the world scheme of things. So when I got to MIT and set up my own lab, was about the time that these microscopes called fluorescent microscopes were invented, where you could shine blue light on a cell, and the pigments, the chlorophyll pigments that the cells use to photosynthesize, turn red. And this is a picture of a bunch of phytoplankton fluorescing red. They emit the red light. And that was tremendously exciting and opened up all kinds of possibilities for viewing these things. Well, at the same time, there was an instrument being used in biomedical research called the flow cytometer that I'll show you here, that where you could inject a sample into this fluid stream, and these are individual cells. There's a tiny little capillary tube, so they go single file through this, and you shine a laser beam on that. And as each cell passes through the laser beam, you can collect the fluorescence from the cell, and you can also collect the light that it scatters, which is a function of cell size. And this is an instrument that's used in hospitals and biomedical research. But we thought, wow, this could be really exciting for oceanography, and wondered what could happen if you just injected seawater through this, and all the different phytoplankton could be imaged. And so we tried that and took this instrument to sea. There. And as you can see, this is a oceanographic cruise, a very rough one. And we actually didn't expect it to work that well, because you're focusing this tiny little laser on this tiny little stream, and you have to keep it focused. And here's where I have to give credit to Rob Olson. Karin gave me credit for this whole Prochlorococcus story, which is very generous, but the discovery was a team, and Rob Olson was a postdoc in my lab, and he was very adept at working with instrumentation, and was able to make this thing work on a ship, which made the discovery possible, actually. There was a whole team of us. So it worked. And what we noticed was there were these tiny, tiny, tiny little cells that were emitting red fluorescence that were a size that nobody imagined a photosynthetic cell could be. And here's a picture that was taken later. And these are two little Prochlorococcus cells, fluorescing red. And they're less than a micron in diameter. So if you lined up, 100 Prochlorococcus cells here, that would be the size of a human hair. And here's a picture of those same two cells under a regular microscope. So you can see why nobody knew they existed. They just look like all the dirt and specks on the microscope slide. So this was really exciting. And it took us a while to appreciate how exciting it was. And we and others studied the distribution of these cells and found that they were extremely abundant in these very large open ocean gyres. This is a satellite image with false color, but the blue is where you don't have very many phytoplankton. And then the red is along these coastal areas where you can have the blooms that Karin was talking about, of sometimes the bad phytoplankton that get in the way of our human activities. But out in the open ocean, you don't have those blooms. They're very low nutrients areas, and Prochlorococcus dominates here. The water there is incredibly blue. Looks like people often refer to this as ocean deserts, as if there's no life there, but they're teeming with Prochlorococcus. There's 100,000 million per liter, 3 octillion, that's 10 to the 27th on the global ocean. The total weight of Prochlorococcus is equal to 2 times the human population. And here they are in a culture showing you their little round green balls. This is just a regular microscope. And here they are in a dense culture showing the beautiful green of the chlorophyll in the culture. So meanwhile, I was teaching introductory biology at MIT at the time, and I came across this commencement speech by David Foster Wallace at Kenyon College. I don't remember why I was reading it. It was a very moving commencement speech. And he started it with a parable describing a big fish swimming through the ocean, sees a bunch of little fish. I'm paraphrasing here. And he says, "Morning, kids, how's the water?" And the fish swim on. One of them turns around, looks at the other, and he says, "What the heck is water?" And the purpose of this speech was about the human condition and going through life, not really recognizing what's important. And it was all about compassion and humans and how we should live our lives. But to me, as a nerd, it suddenly said, we don't pay any attention to life on Earth and how it functions. And most importantly, the importance of photosynthesis. And David Foster Wallace says the most obvious, ubiquitous, important realities are often the ones that are hardest to see. And that's the way I feel about photosynthesis on the planet. So I'd like to just talk a little bit about that before I get on to Prochlorococcus. So I made this slide for my biology class because to me, it's life on Earth in a nutshell. It's the most important process on Earth, which is plants and phytoplankton here take carbon dioxide from the air and water and sunlight, and they create oxygen and carbohydrates, living biomass, sugar. And it's that biomass and the energy in the bonds of that biomass that runs the rest of the planet, the rest of life on planet. So we and other organisms take that and we basically burn it in respiration, emitting carbon dioxide, and use that chemical energy for being alive. But how often do we think about the fact that the warmth of our body is actually literally solar energy, and that the pumping of our heart is possible because of the solar energy? that's been processed through plants. Plants make it possible for us to get that energy. But that's where it comes from. The other thing is, how often do we think about the mass of our bodies being made from carbon dioxide? And the mass of all of life on Earth comes from the air. And this is such a profound concept that I now, when I walk through life, well, if I see a tree or any kind of green thing, I'm constantly reminded that that's what's making it possible for us to be alive. So there's a famous study, and if you go on YouTube and ask where does the mass of a tree come from, and you see people being interviewed. In this case, the researchers interviewed graduates of MIT on graduation day. And they asked them, they said, here's a seed, you plant a seed, it becomes a tree, you cut it down to make firewood. And they asked, where did the weight of the logs come from? And the students couldn't answer that. And this is like a profound concept that all of life on Earth is made out of air. And I encourage you to go to YouTube and you can look up these. There's a whole bunch of them, not just of MIT graduates, but also just people interviewing anyone on the street. Or ask your friends, where does the weight of a log come from? And where does the weight in your body come from? And see what kind of answers you get. Okay. So photosynthesis is just an extraordinary thing. And plants on Earth fix about 50 billion tons of carbon annually using this process, making all of this biomass. But half of the photosynthesis on Earth occurs in aquatic ecosystems, mostly in the ocean. And that's just astounding when you compare the biomass, the actual weight of the total amount of plants on land versus the total amount of phytoplankton in the sea. It's a tiny little fraction in terms of biomass, but they're lean little photosynthesis machines. They're just amazing because all they do is photosynthesize, photosynthesize, and divide. They don't have trunks or anything like that to maintain. So pictures like this, textbook pictures, really give you the wrong image of how it works. And so I'm showing, this picture is from one of my children's books that I'll show you in a minute. But really, in the oceans, the phytoplankton are just this thin little film of photosynthetic biomass on the top of about 200 meters where the sunlight penetrates. And then you have this huge, deep, dark ocean. So you have this photosynthesis is feeding the entire ocean system. And some of the organisms swim up, at night to feed, and others wait for stuff to settle down. Most of what's photosynthesized in the surface ocean is then respired back to CO2. But as I said, some of it settles down to the deep ocean. And you have organisms down here that are consuming it and respiring and regenerating this carbon dioxide in the deep ocean where there is no photosynthesis. So there's no commensurate production of oxygen or consumption of CO2. So you end up with this elevated concentration of carbon dioxide in the deep ocean. And this is referred to as the biological pump, this constant rain of organic carbon being regenerated in the deep ocean. And it's an integral part of the Earth system. If the oceans were dead and this did not occur and all this carbon dioxide mixed into the atmosphere, it would be 2 to 3 times the concentration in the atmosphere. So phytoplankton are playing a critical role in keeping CO2 at high concentrations in the deep ocean. And that's another role that's unappreciated. Okay, using this as a framework of life on Earth, I call this Life on Earth Abridged. This is really all you need to know. It's what everybody should know. We can superimpose on this what we humans have done to this system, and that is we've dug up the fossil fuel, this piles of coal, are really just fossil photosynthesis. It's products of plants that got fossilized and were not respired in the day, and now we're digging them up and we're burning them. And burning is exactly the same process as our biochemical respiration. We're using oxygen to break down this organic carbon to release energy, in this case, to improve the human condition for our human well-being. And that has completely changed life of humans on Earth. And doing this just over a few hundred years, when it took millennia to bury this fossil fuel, the CO2 in the atmosphere has started to increase dramatically. And this is our global experiment, which I'm sure you've all seen many times, this graph, which is a time course of atmospheric carbon dioxide, in this case going from 1960. These seasonal cycles can be seen here each year. The Earth, you can actually see the Earth breathing. And it shows there's been a 20% increase since 1990, which is an incredibly significant change in the Earth system. And people do argue about global change and global warming, but you can't argue with this data. It's a dramatic change in CO2. And I felt so passionate about photosynthesis and life on Earth that I teamed up with Molly Bang, who's a friend and a very successful children's book author. And we've written this series called the Sunlight Series, which describes basically most of what I just talked to you about. They're written for children, but I really was thinking of everybody when I wrote them. And this one's about photosynthesis on land. This one's about photosynthesis in the oceans. This is about fossil fuel and how it was deposited over the years, called buried sunlight. And this one's about global circulation of water and how that works. Okay, so that's the big picture. That's what Prochlorococcus inspired me to think about. But let's go back to the star of the show, which is Prochlorococcus. Its photosynthesis globally is equal to that of all the crops on land shown here in this map. And this makes you wonder, how could a single species be so productive? How could it do so much? You'd think that would be a very unstable system to have a single species be that important in the global photosynthesis. And what we learned in studying it over the years, that it's not one thing. It's, even though all the cells are very closely related evolutionarily, they have different functions in the system. And we learned that there are ecotypes, and that the cells, for example, on the surface layer of the ocean are more adapted to high light intensities. The cells down here can photosynthesize at extremely low light intensities. In fact, they're the most efficient photosynthetic machine that exists on the planet. They're extremely efficient. And then there are some in the middle that are actually adapted for the mixing, the changing light intensities. And then we also learned that if you look at temperature gradients in the ocean, this cartoon picture shows you that the ocean waters are much warmer along the equator. And we found that those cells are adapted to higher temperatures than these cells that dominate at the higher latitudes. So then genomics entered the picture. We were fortunate enough that Prochlorococcus was one of the first microbial genomes to be sequenced. And that was tremendously exciting. And it opened up a whole new world of study for us. And eventually we were able to sequence the genomes of individual cells by using that same flow cytometry technology that I showed you. You can actually sort out droplets that have individual Prochlorococcus cells in them, and you can sequence their individual genomes. And so one by one, we could pull them out of ocean samples, and we found that each cell had about 1,700 to 2,700 genes. They had different sized genomes, which was very surprising to us, since they all look exactly alike. But what we found as we sequenced more and more, that they only had about 1,000 of those genes in common, and that each one time we sequenced, we would discover new genes that Prochlorococcus had. And a theoretical projection says that they have 80, the collective, all the Prochlorococcus in the oceans have about 80,000 unique genes, and that is 4 times the size of the human genome, 4 times the amount of information as we have in our genome in this tiny little microbe. So it's not just like these genetic differences. We know, I mean, we all have exactly the same genes, but we have different variants. So we have different eye colors among humans. The variability in Prochlorococcus is more like this. If some of us could live underwater, some of us could photosynthesize, some of us could fly. This is exaggerated, but to give you the idea of the different functions of these genes. So in trying to explain this in a number of lectures, I asked one of my students back in the day, we need an analogy for this. And one of them came up with the smartphone. If the cell is a smartphone, and then each of the apps are a gene, we can think about it in a way that is analogous to this diversity of genes in Prochlorococcus. For example, all the smartphones come with a certain set of core genes in quote. They all have that. In the old days, those are the ones that you couldn't even delete, but now you can. And then the phone is made up of different apps. So these would be analogous to the core genes. And then you can think of the specific types of apps for different functions as niche defining genes, things that define how that microbe would thrive in a certain environment. For example, this person would be a travel enthusiast because they have all these travel related apps. You might find somebody who's into financial dealings. They have all these financial apps. Or this person is into weather. Their job depends on weather. They have a lot of weather apps. So if I collected all of your smartphones and looked at the apps that you use most frequently, I could say something about how you live your life, your niche. So with all the genes that are available, or apps that could be put together to assemble this organism, the diversity possibilities are absolutely endless. And this is what we see in Prochlorococcus, just this extraordinary genetic diversity. And we've come to think about it as a superorganism. And it's this diversity that allows it to occupy the upper 200 meters of the open ocean in great abundance. So at the same time, as I'm pondering this whole system, I'm constantly reminded that this tiny little cell with these 1,000 core genes, the information can do something that we humans have not figured out how to do. And if we could figure out how to do it, our whole problem with energy would be solved. So with the information in 1,000 genes, this tiny cell can take sunlight, CO2, and nothing but inorganic nutrients. Doesn't need any, or vitamins or anything that any other cell has made. And it can make biomass, chemical energy. And I'm constantly inspired by this and humbled by this. One day we'll figure it out, and some people are trying to use Prochlorococcus as a chassis for building artificial photosynthesis, but it hasn't been done yet. So that's a big, big challenge. Okay, so Prochlorococcus has also taught us about Earth history a little bit. It got me interested in studying. In sequencing the genomes, you can understand something and comparing them with other organisms, you can begin to understand something about evolution in the past. So if we look at, this is what the Earth actually looked like about 600 million years ago, which is when we think Prochlorococcus first appeared, in its ancient relative first appeared on the Earth. And of course, here is what the Earth looks like now. This is just to remind us about how dramatic our planet changes over timescales like this. And I'm not giving Prochlorococcus credit for this geological shift, but this is the world that it lived through as it was evolving. Looking at a much longer time frame, we're reminded that looking at atmospheric oxygen on Earth, that for billions of years, the oxygen concentration was extremely low. We were an anoxic planet. And then there was this great oxygenation event when photosynthesis evolved. Again, if it hadn't been for that, nothing would be the way it is today. We'd probably still be a microbial planet. So this was the beginning of photosynthesis evolving. And then you have this big burst here where it went up to 21% in the atmosphere. And this is the period when Prochlorococcus evolved. And again, I'm not giving Prochlorococcus total credit for this bump in oxygen, but we think it played a role, its evolution played a role in bringing the oxygen level up to where it is today. And this is also the period of what is called the Cambrian explosion, when there was an incredible explosion of diversification of life on the planet, which is, given credit for the ultimate movement of life, and explosion of life that led to who we have on the planet now. And we can even see in Prochlorococcus's genes, they acquired genes for breaking down chitin, which is a compound that's in sort of invertebrate organisms, about the same time of this Cambrian explosion. So it's very exciting for people who study geobiology that we collaborate with to look at Prochlorococcus's genes to see what genes they acquired back during this period. And that tells them something about the timing of things. This is not something we work on, but we work on it in collaboration. And I always love learning about stories that Prochlorococcus can tell. So what does the future hold for Prochlorococcus? The Earth is warming and the oceans are warming. And as I told you, there are these high temperature adapted Prochlorococcus cells that we project, the models project, that they will thrive in the warmer ocean. And the models project that there'll be a 30% increase in Prochlorococcus over the next century, because they're very well suited for the warm temperatures and the highly stratified waters and low nutrients that are projected to occur then. And of course, this is great for Prochlorococcus, but this is not great for the entire phytoplankton community. So that would change the structure of the food web, which would undoubtedly be not beneficial for global fisheries. Okay, just to wrap up, and Karin mentioned this, and I was very touched by the way you described how we study Prochlorococcus, because it really captures what this organism has taught me about how to study life on the planet. And I'm always taken by this quote by John Muir, who's a famous naturalist, was behind all the national parks in the United States. He said, when we try to pick out anything by itself, we find it hitched to everything else in the universe. And as you can tell by my very brief story of my journey with Prochlorococcus, that's what's happened. So if I think of Prochlorococcus as my muse, it's taken me on all these journeys genomics. We study the proteins, diversity that it makes, cell metabolism we study. I didn't have a chance to talk about this, but we study the viruses that infect it, and we're trying to understand what eats it. This is one of the bigger challenges right now, is because the abundance of Prochlorococcus in the oceans stays relatively constant. So they double once every 1 or 2 days, but something is eating them at that same rate. And this is really difficult to study, and we're trying to get to the bottom of that. We also study them. There's time series stations off Bermuda and Hawaii, where they love to live, which is very fortunate for us. And we study them, how they change with the seasons there. And of course, we're always interested in projecting their effect on the global biosphere. So this one tiny little cell has taught me so much and forced me to learn so many different fields in biology. And I've been fortunate to have incredibly talented students and postdocs who are experts in all these different fields, and an incredibly diverse group of people that contribute to this. Okay, just to wrap up. So what has Prochlorococcus taught me about life? Just in a summary, as I just said, everything is connected. So, whatever you decide to study, as they say, pick a lane and swim like hell. And then you'll find that even though you're focused on a particular lane, there are connections that will really bring you into exciting different areas. And also, it taught me how much diversity matters. And we all know this, but the fact that this tiny cell can dominate the oceans is simply because of the diversity of its functions. It's also taught me to expect the unexpected. And, I was so lucky to be part of this team to discover Prochlorococcus. So I always credit the team, not me. But I will take credit for focusing on the unexpected. For the first 5 years that we studied it, we couldn't get research funding for it. And some people even denied that it existed because you couldn't see it if you didn't have a flow cytometer. But yeah, I just knew this cell spoke to me. It just, it said that if I stuck with it, that it would give me many, many, many paths to study. So I did. And so I was very lucky. And my other sort of life lesson was to be guided by gratitude. I feel so fortunate and lucky to have happened upon this little cell that has really become my life. I mean, this tiny little cell has given me my career. And I'm incredibly grateful for that. But most importantly, too, I'm so grateful for the incredible students and postdocs, one of whom is on the panel today. Very excited to see you again, Olof, over the years, who are really the ones that have made all of this possible. As I said, I am not an expert in all the fields that we've gotten into, genomics, proteomics, my goodness. But I had to work up the courage to sort of be stupid in order to take my lab in those directions. And that courage came from the expertise of these extraordinary people. And also all of the funding agencies who allowed us to wonder, to just do basic research, not... Most people, if I talk about Prochlorococcus, they say, well, that's all great, but how can we use them? And I say, well, they're out there doing their job in the ecosystem. We don't need to use them. And I've been so lucky with funding agencies that are willing to just let us follow the cells, follow what we learn. And of course, I have the deepest gratitude for the Crafoord Foundation, which gave me this incredible prize, which inspired so many people in our field that that phytoplankton were recognized as warranting this kind of respect. So I'm so grateful for me to have received the prize, but for the entire field, I mean, it was an incredible boost to be recognized in this way. So with that, I say thank you very much, and I look forward to the panel discussion.
Thank you very much, Penny, for that wonderful lecture and sharing your story and the story of Prochlorococcus. So, welcome back to the panel discussion. And of course, you've already met Penny over in Boston. But here with me in Lund, I also have a panel. And there are 3 participants. And here closest to me, Dr. Hannah Blossom. Hannah is a PhD in marine ecology, and she has focused on phytoplankton ecology, just like Penny, especially those nasty phytoplankton that cause harmful algal blooms. Then next in line, we have Associate Professor Olof Berglund. He's also one of Penny's former postdocs, so he worked in her lab. And he's a professor in ecotoxicology, so he knows a lot about pollution and the effect of all different kinds of pharmaceuticals, all sorts of things, and metals on phytoplankton and other aquatic organisms. And furthest away from me, we have Professor Karin Legrand. She's a professor of marine ecology at Linnæus University. So I forgot to say the other 2 are from Lund University. But Karin is from Linnæus University in Kalmar. And she has also focused on phytoplankton in her research, especially interactions between phytoplankton and other microbes. But also she has done a lot of work with using phytoplankton to help society deal with many of its problems. So she can probably answer some of those questions that you might have. So welcome, panel. Welcome back, Penny. So I have some questions that are popping up here on Menti. I will start with you, Penny, because I got a question here regarding scientific naming. So the question is, why such a complicated name for the smallest phytoplankton?
Well, that's a very good question and also very controversial. In science, naming has historically been a very important thing and a way to keep things identified. And I honestly don't know the history of why things are named the way they are, but they have Latin or Greek roots. And when we found this organism, I went to John Waterbury, who was one of our collaborators, who is an expert on taxonomy, it's called. And he came up with the name Prochlorococcus. Prochloro meaning before chloroplasts, and coccus meaning little round thing. That's what these roots mean. And it turns out that now that we know what it is, it's not the right name. It's not. We thought it was related to higher plant chloroplasts, but then when we were sequenced it, it isn't. And yet, and there's been lots of movements to rename it also for other reasons. And, it just complicates things because you have half the literature with a certain name and then you change it. It just makes it impossible. So that's a good question. It would be much easier if its name was like Joe or Susan or something like that. But this is the history of science, and this is how we do things. So we have to keep working within that structure.
So you're stuck with a name then? For
now, we're stuck with it. Somebody may. There are people that have changed the species name actually in the literature and want to separate it into many different names, all of the different ecotypes. There are papers that have described them with different names. And we have resorted to numbers to we just describe them by numbers that have meaning to us, these little finely diversified strains of Prochlorococcus. But the entity Prochlorococcus still maintains its integrity. It's the smallest cell in the ocean that fluoresces red. And no matter how you sequence, when you sequence those cells, they're all very closely related, and you can identify them as Prochlorococcus.
Okay, thank you very much. Another question that we have, and I think I will start with Olof for this one. And the question is, what about sound? Does sound affect how algae grow?
Ooh, that's also a very good question. A little bit outside my topic. We have done some work on sound pollution, but I have not seen any sound pollution in aquatic environment, because of course sound will affect aquatic environment differently than when it travels via air. So sound can pollute areas and could disturb functions of different organisms in air that rely upon sound and or attraction avoidance with sound. But I dare not answer whether they could affect phytoplankton.
Anyone else having any input? Yes, I can see Karin has some ideas about how sound can affect phytoplankton.
Yeah, I don't know actually if it does affect directly phytoplankton, but I know it affects, for instance, marine mammals like dolphins. It does also affect fish in some way, because fish don't like very much noises. And there are many mammals and marine mammals and fish that feed on phytoplankton. So indirectly, I would say that it could be. But as we heard from Penny, Prochlorococcus, that tiny one, is not directly eaten by big fish or big mammals directly. So we don't know that. This would be maybe a subject for further research.
I do know that the sound in organisms that are bioluminescent, sound waves will make them, physical disturbance or sound waves can make them light up their lights. So that could affect things, maybe predators or however they use their bioluminescence could impact, maybe.
Okay.
I guess.
Yeah, thank you. So there are some ideas here. What do you say, Penny?
That's I don't know anything about direct effects, but I agree that everything's connected. So whatever organisms that sound does affect, I'm sure in some way that will affect the phytoplankton. But again, directly, I don't know. That would be a great study for somebody. And I hadn't thought of the bioluminescent thing, but there's a direct effect.
Mm-hmm. Thank you. There's another question specific for Prochlorococcus, and the question is there a type of Prochlorococcus that is most efficient?
Oh, good question. Well, we think these, the ones that live at the bottom of what we call the euphotic zone, which is the zone in the ocean that light will penetrate through to the deep water. It really, below 200 meters, there's not enough photons for photosynthesis. And the cells that live down there are slightly bigger than the ones at the surface. They have bigger genomes, and they can grow at light intensities that the other ones can't make a living. So they're the most efficient, and I hope that somebody someday will study them in great detail and show that their photosynthetic mechanism is astonishing.
So it sounds like there's definitely things to do in the future.
Yeah, plenty.
Here's a more general question that I know has been debated scientifically. So the question is, how does iron affect the productivity of the ocean's phytoplankton? And if it does, could you recreate that effect artificially? And I will let you start, Penny, and then see if someone else in the panel wants to add something.
Well, that's a very good question. And I don't know if the person that asked it I've written several papers on this. And it has been proposed that we know that iron does limit photosynthesis in regions of the oceans. And some people have proposed that we should artificially fertilize those regions to accelerate that biological pump that I talked about. And my feeling is that it's more than a feeling. The models have shown that even if you fertilize the entire ocean, that it would not change significantly the trajectory of CO2. But more importantly, if you do that, you change the food web dramatically. I mean, that would be by design, because there's only certain species of phytoplankton that bloom when you add nutrients. So you have to be prepared to do that, which would change everything all the way up the food web. And Karin pointed out these algal blooms in coastal waters that are caused by excess nutrients that cause these blooms that, if they get thick enough, can cause anoxia in the water. The carbon of the phytoplankton gets metabolized by bacteria. They consume oxygen, and oxygen concentrations go down, and you have massive fish kills because the organisms can't survive at those low oxygen concentrations. If you fertilize the oceans systematically and a large scale, somewhere you will have these low oxygen concentrations. And there are species of bacteria that thrive under those conditions, and they emit greenhouse gases like methane or nitrous oxide that are much more powerful in terms of the greenhouse effect than carbon dioxide. So there are many indirect, again, it gets down to everything's connected. So you do that, and there are many indirect effects. So it's a provocative proposal, and there are people who are still pushing it. And in fact, there's an entrepreneur who actually did a unauthorized fertilization experiment off British Columbia some years ago. That's a long story. Whoever asked that question, if you want to email me, I can give you all kinds of information. So, good question.
Thank you very much, Penny. Anyone in the panel want to add anything?
Yeah, not on that topic per se, but I think in general, when you want to try to remedy effects from human activity by introducing another human activity, you may want to be careful what you're doing. Perhaps it's better to try to reduce the first activity to remediate those kind of effects. Because as Penny mentions, everything is connected, so you don't really know what you're starting if you consider a new activity to remedy the first one. You have to think it through carefully.
Yeah, good point. Karin?
Yes, I have a comment to follow up on both Penny and Olof's comments here. In the Baltic Sea, for instance, you have shown, Karin, earlier that some of the algal blooms were those nasty filamentous cyanobacteria in the summer that tend to extend due to eutrophication, warmer temperature, and so forth. And research has shown that if you add iron, they are the ones who will benefit the most of it. So it's exactly typical example of you add a remedy which becomes an anti-medicine. So no, it's probably not the way to go, adding iron. And my other comment on the iron debate, since I was a young researcher, that question has popped up in the literature, and I have to say that I give a lot of credits to scientists like Penny, who took the discussion and the debate to another level than just the discussion between among scientists, but also took the discussion with politicians. And there are a few other examples than just the iron question, but this is very important that when you become a scientist, you need to think that you are also a concerned citizen and that your voice matters. And Penny showed that very much and inspired a lot of us to actually follow in those steps to take the discussion with decision makers.
Very good point. What is our responsibility as scientists? And here I can just add to the students out there that you can still send in questions. And so I thought we would move a little bit away from Prochlorococcus and more to science and research in general. And so you might have questions on that as well. And I think this is a question I want to address to several of the people in the panel. And it's when you, a researcher, present scientific results to a general and diverse audience, how do you prevent against the misuse of your scientific research? Penny, do you want to start?
Boy, that's a good question. I think the iron fertilization example is a good one, actually. People that study phytoplankton study nutrient limitation and what causes it. And in fact, we were on one of the first scientific iron fertilization experiments in the ocean. Because there was a debate early in the early days whether iron actually was limiting, and John Martin, who's the first person that really pushed the fact that iron is limiting, designed this small-scale scientific experiment with just scientists studying phytoplankton and their response to iron. And I was interested in, is iron limiting Prochlorococcus? So we went on that first cruise. And so I experienced firsthand how the press can misinterpret scientific research. And so that cruise was interpreted as an experiment in what's called geoengineering, where were we studying iron as a mechanism for sequestering carbon? And I certainly wasn't. And the cruise was basic research, but because this hypothesis about geoengineering was out there, it was interpreted otherwise. And so I think that's partially what moved me to write articles about the risks. And I agree so much with what Olof said about, that oftentimes human solutions to problems we created end up creating more problems. We know what causes global warming, and we know what we need to do to stop doing that. So I think we should focus on not doing things that are causing harm rather than trying to fix them by doing something else.
Mm. Thank you. Karin, do you want to answer that question as well?
Yeah, I would like to say that my experience in talking to school pupils, students at the university, and then decision makers is to stick to my message, to what my science is saying, as much as I can interpret it, but not speculate to the moon and back. I can probably speculate to Olof's table, because this is how you research, having ideas and try things, but definitely stick to what your evidence is showing. And this is why today the climate scenarios, for instance, we have spent so long to kind of fine-tune them, and still we are not completely there. And we know it's warming up. This is a fact. How much is it in which part of the world? We are still researching on it.
And this is totally okay. Stick to the message.
Mm-hmm. That's your message. Olof, do you have any comments?
Yeah, I don't know whether the research will be misused. I think, as Penny mentioned, it may be misinterpreted and maybe taken out of context, and that's a problem, of course. And then it's important that you try as hard as you can to not complicate things that are able to misinterpret. But misuse, I think, that's a different thing, because then you actually actively misinterpret something if you misuse them. But misinterpretation is common, of course, and often that comes from taking things out of context and applying them in a domain where they're not applicable. And that, of course, relates to Katrin, that, well, don't speculate wildly. Do speculate, because that's good fun, but stick to your domain if it's a sensitive issue that you suspect could be actively misinterpreted, which is often the case in ecotoxicology and pollutants, of course.
Yeah.
Anna, what have you experienced?
Well, I was just thinking about how we are taught how to write papers and present our results, but science communication to different audiences is not something that we're really taught to do or how to do it well. So that's something maybe for the future, or that could be better for, I don't know, the younger generation that are more good at social media and all that too, that there needs to be some science communicators also working with the researchers. Maybe that would help. I don't know, but
so science communicator is a future profession, maybe.
I think it's picking up, yeah.
Yeah, and so that actually brings me to another question I have here, and it says, I have no idea what profession to go into, what to study. How did you find your way? Maybe we can start with Penny.
Oh, I always tell students, I know that this is a tough decision, and I had no goals when I went to college or all the way along. I would just say, find something that you're interested in. Find something that interests you. And if you just take steps in that direction, it will lead you somewhere. And I know a lot of my students worry about, what if, and I don't know exactly what I want to do. And I say, you have no idea how your life is going to unfold. Everything changes. And so the most important thing is finding something you're interested in. And for some people, that's a little difficult because they're interested in a lot of different things. And that's where you just pick a lane, and you go with it. So my advice is always, don't worry about it too much. Find something you're interested in and follow that, and it will serve you well.
Thank you. Hannah, is this also your experience?
Yeah, I think that for my own experience, I guess I wanted to be a marine biologist after, in my high school, we were fortunate enough to have trips that were offered, and one of the trips that I could go on was to the Galapagos, and we went with our biology teachers, and that was just so inspiring. And I think, and ever since then, almost every year, I just wanted to do a trip or to, volunteer or get experiences and do internships and just try everything out. I mean, I think also, you do have plenty of time. I feel like I'm just starting on my career, and I graduated high school 17 years ago. So you have plenty of time to figure it out, I think, as high schoolers. And my advice would just be to take every opportunity and to travel and do things like internships and stay connected with the people that you meet. And you will figure it out. You'll get inspired somewhere.
Thank you. Olof.
Yeah, I agree. Find something interesting and stick to it, and also perhaps for me, it's also finding people that I find interesting and I can communicate with and can inspire me as a student and then as a PhD student. I picked mentors or supervisors more for the people they were or how we got along than the topic itself, because once you start, everything will get interesting after a while. But I will also recommend maybe to pick some mentors or people that you get along with and that are pleasant to work with.
Karin?
So after Olof's seriousness was pick your supervisors, because I was thinking to advise, yes, if you want to become a scientist or you wonder, just contact a scientist and talk to them, and then you will see how you feel, get an internship, exactly as you said. But then I realized, you need to have the interest. You need to work with people that are really interesting and cool, and you get along. Always about interactions, but you need to have fun. Because science is about being curious, but you can't continue to be curious if you don't have fun. So that's my advice. Find something you are interested in, stick to it, work with the right people, but don't forget to have fun, because this is the base to have a good thinking.
Yeah, and because I think also, you do need to be passionate about it, and so if you're having fun, you're going to find your passion, and you do need that to continue in science, I think.
Yep.
I'd like to just add to that, say that, I started out as a math major in college, but I didn't resonate with my math teacher, and I realized I wasn't going to be very good at it. And so I liked my biology teacher, so I switched to biology. And then I got interested in chemistry, but, so I think all of those factors that you're talking about. And then I had fun on the lakes and, going out and doing the field work. Those are all really key variables, what you're interested in, the people you interact with, and how much fun it is. So.
Yeah, thank you for making that point, Penny, that you can actually change your mind. I also did. I started with art school and did not find my people there and switched to biology, and that's where I found my group. So absolutely, you don't have to get on the right track right away. You can move around.
No, I started as a clear mind. I will be a space engineer. And I'm studying phytoplankton now, so, it's all about change.
It is. So, and I think this connects nicely to the next question, which is, what is the best and the worst part about being a researcher? What do you say to that, Penny? The best and the worst?
Well, the best part is the thrill. I always say, Prochlorococcus is like opening a present every week. Somebody figures something new out. But that's the best part. But the best part is also interacting with these incredible, talented students who are opening the present and showing it to me. So that's what keeps me going. The worst part, this depends on where you are and what country you're in and how the worst part is the constant pressure to raise funding to do the research. In some countries, the way science is structured, and so I always feel like I'm running a small business. And I have these dependents in the lab who are students who I support and postdocs that I support. But again, I've been incredibly lucky by getting support from a lot of it from foundations that recognize the importance of basic research. So I feel very lucky. So the benefits definitely outweigh any of the pressures, and that's not something to worry about.
Mm-hmm. Thank you. Hannah, what's your take?
It's similar that the best part is, the thrill of the discoveries and not major, it doesn't have to be major discoveries, but any result from an experiment can be so exciting. and you're always learning something new, and so it's never boring. That's the best. You just always learn something new. Make new little discoveries. And those little discoveries, are more exciting than the bad parts are bad. But I agree that trying to get funding is hard, and you need to be able to handle getting rejected and, having some failures along the way. That's part of also experiments and doing the research, is that you'll get some failures, but then the little discoveries will go further than the failures take you back. So it's important to just accept that, and but it can be hard when it happens.
Yep. Olof?
Yeah, I can only agree. I have a hard time finding what's bad with being a researcher. Of course, getting funded and pressure, but I think that's similar to all kinds of jobs. The positives outweigh. It's a fantastic line of work. I'm almost amazed I get paid to do it. You get to settle your curiosity every day, and you get to teach. Because the students, I think, is the highlight for me, both graduate and undergraduate students. Because that keeps me up when maybe the success is not always there in the research. I know I'm doing something because I'm educating, and that's a big part of being a researcher as well, I think.
Following up on, I agree with everything has been said, and this is what happened when you are the last to comment. But I want to just make a point to the students here. You don't need to study science with the goal to be a scientist, as we all are here, professional scientists, because your experience with studying anything related to science, you will become critical, you will become synthetic, you will learn so many different things that will help you in any kind of jobs you choose in the future. So don't hinder yourself to study science and be with scientists for a while during your studies, thinking that, maybe you don't want to be a scientist, or maybe you do. It doesn't matter. Just give it a try, because this is something that you keep for, your entire life. It's a way of living. That's the best with being a scientist. And then the worst, in my case, I would say that, unfortunately, Penny, I don't know if you have been like that, but I am seasick so much that I had to use like special drugs to be on cruises, but I enjoyed those cruises tremendously. But God knows I was seasick. Woohoo!
Yeah, that's the first question I ask if students come to me and say, I want to be an oceanographer. I say, have you been on a ferry boat? Do you know whether you get seasick? But there are some drugs that, I know some oceanographers, lifers, that get horribly seasick, but they still take the drugs and go. But you're right, that is a downside.
It was a little downside.
So now over to something a bit more specific. So this is a question, and it says, in school we are asked to be careful about controlling sources and making reference lists when they do, when you write projects and essays and so forth. How do you know if a source is reliable? Penny.
Wow, that is such a good question. Because that is such an important feature of our work, and really getting to the first primary reference, and it's something that is, I'm sure that it's taught in schools now, but because you can just go to Google and Google something up, and you get all kinds of stuff. But there are scientific journals that you learn to peer. First of all, the most important thing is that it comes from a peer-reviewed. If we're talking about science, the most important thing is that it comes from a peer-reviewed journal. That is, in science, that is the way we have checks and balances in. And it's a pretty amazing system when you think about it. Is that in order to publish a paper, it has to be reviewed by, 2 or 3 peers, and that's how we keep the quality control. So if you're talking about science, it definitely has to be a peer-reviewed journal. Beyond that, boy, I don't have good advice, but maybe somebody else on the panel does.
Okay, thank you. Let's hear the panel. Katrin, do you want to comment?
Yeah, I was, of course, thinking about peer-reviewed as a main thing. Today, there are also places, repository, where you can publish your first data, your drafts, and you get comments from the whole community before you get it published. But usually this is based on trust from the peers and their rigorous review from each work. I think that's the reliability that I can think of, and not going for any kind of journals, just the peer-reviewed ones. Then this usually is a quality check, but this is a very difficult thing.
And how do you know as a high school student if a journal is peer-reviewed?
Well, as a high school student, I would say that you have teachers and supervisors that are there to guide you. I just kind of think as teachers, we usually give a very thorough introduction to our students about the library, about how to work with sources, as with journals, etc., in the scientific community. I think you can maybe continue to comment on that, Olof, how we do that.
Yeah, that work, I agree. Peer review is important, and also the primary source of this finding or results, because along the way sometimes people get a little bit lazy and cite something, and that may spread, but the primary source maybe said something else. From the scientific information, that's usually quite easy to find out. Is it peer-reviewed? You can look at the journal's homepage. It usually says so. But also you might consider that you may want to know whether there's sort of a scientific consensus on this view or result, because if you look for something in scientific papers, you can probably find it. Somebody in some study had found something. But just one finding is maybe not a scientific consensus. You may want to look around to see whether this was just a random result from 1,000 studies and 999 others say another thing. So that is also a way to weigh the evidence back and forth in an issue.
Thank you. Hannah, do you have anything additional to add?
I don't think I have anything additional to add.
Yeah. Mm. So it's possible, but it's not easy, and I think it's a very good point to remember not to cite somebody that has cited something else, but actually go back to the original first source. That's really important. So thank you for mentioning that. So speaking of scientific results and consensus, here's another question. What do you do to avoid any ideological or, I suppose, political influence upon your scientific research? Penny, do you have a comment? Answer?
Well, I guess that kind of brings me back to the whole iron thing. As I said, I've written 2 articles on the potential side effects of large-scale ocean fertilization, and what motivated me to do that was seeing the direction that interpretations were going in terms of what is called geoengineering now. So I think, as we've already said, I think the thing, if you see science being used in a way that could lead us in a harmful direction, I think it's important to speak out. It used to be, we believe that science is not advocacy, or active. Science is not activism. Science is objective, but we can also sort of carefully point out the scientific understanding as we understand it. And I think it's really important that people understand that our understanding is constantly evolving, especially when you're talking about ecology and the interactions in the Earth system. So I think there are ways to speak out without taking a political position or whatever, but just explaining our understanding of the system and what could go wrong through these perturbations.
Thank you. Anyone in the panel like to comment?
Yeah, I could comment on the fact that I'm not completely sure how to answer that question, but I think it's super important again to our job is to be scientists, and we can provide politicians, we can provide the society with different scenarios, with different options, hypotheses that we can deduct from our data and our results and our discoveries. But we are not there to decide. We are there to recommend. We need to take a step to take our responsibilities. And yes, it's a constant, as Penny said, it's the constant change. We have reviewed paradigms during the past 200 years in science. And that's very important to keep that open mind. Because sometimes the one study out of the 1999 is the one that will bring Prochlorococcus to reality. For instance, or mixotrophy in dinoflagellates, which has been denied for a long time. So there are plenty of things that we need to keep this open mind, but we are there to recommend, not make decisions.
Yeah. And I think there's a responsibility that scientists have to point out if something is misrepresented or something, and also that we are transparent about our work and that everything should be clear and reproducible and stuff like that, so that the public can trust scientists and that there's not a mistrust. And then it's a little bit different, but something like that.
Olof?
I can only agree. It's tricky, perhaps not to be political. Everything seems to be politics nowadays, but as long as we explain the scientific methods and the uncertainty associated with our recent findings, I think that's pretty much as far as we can go, and try to be humble.
Yeah, absolutely. Yeah, thank you. One final, more fun question, maybe. So, is it possible to make gummy bears out of Prochlorococcus? What do you think, Penny?
Well, we've never tried that, but theoretically it should be possible because they're pure sugar. And the first product of photosynthesis is sugar. So I think that would be possible. But if you want to make them out of a phytoplankton, you might choose one that's bigger and grows a little faster.
And they would definitely be green, right?
Yeah, definitely.
Any other ideas in the panel?
They will be really tiny, tiny ones. So maybe this is a good, healthy gummy bear.
Yeah.
Okay, well, thank you very much. And on that note, that science should be fun, research should be fun, and this is what keeps us here. I would like to thank the panel for coming to this event and sharing your ideas and thoughts. And most of all, of course, Penny, all the way from Boston, for agreeing to give your lecture here and share your experience with us and with all the students out here in the country. And so with that, I would like to leave the closing remarks to Ove Eriksson in Stockholm.
Thank you, Karin. At least I have had great fun during these hours, so that has been fulfilled, certainly. But before closing this lecture, I would just like to, on behalf of the Royal Swedish Academy of Sciences, to thank Penny Chisholm for an excellent and inspiring lecture, and for coming up early in the morning to have it for us. We are very grateful. And to Karin, Rengefors, and the panel, and also the crew in Lund, who has managed to handle this lecture. And to the Crafoord Foundation for making this whole event possible. And then finally to all of you who have listened to this lecture. So thank you very much, and goodbye.
Goodbye.
Thank you all very much. I so appreciate this opportunity.
Thank you, Penny.